Hello, everyone. My name is Basa Khan. I'm a PGY6 neurosurgery resident at Baylor College of Medicine in Houston, Texas, and I'll be presenting some of the work I did in Dr. Akash Patel's lab on the loss of CDKN2A and B in meningioma. So the main ideas of this talk are going to be that heterozygous as well as homozygous loss of these genes should be diagnostic for grade 3 designation in the upcoming WHO criteria, and that these losses occur almost entirely in our meningioma group C, the aggressive group of tumors that we've molecularly defined. And these findings we published recently in Acta Neuropathologica. So the first point, heterozygous CDKN2A and or B loss should also be diagnostic for WHO grade 3. As you may or may not know, in 2021, the WHO added homozygous loss of CDKN2A and or B to diagnostic criteria for grade 3 meningioma, meaning that even if you have a benign histopathology, if you have these gene losses, you automatically qualify for grade 3 designation. These two genes are located next to each other on chromosome 9p, and they're tumor suppressors whose loss is known to be associated with worse recurrence-free survival in meningioma. A little bit of the background biology. The protein products of these genes are P16 and P15, both of which are tumor suppressor proteins that regulate the progression of G1 to S phase in mitosis. So loss of these genes leads to loss of these protein products and unregulated mitosis in the cells. Previously, the largest series was from Heidelberg, and they showed that homozygous loss of these genes predicted worse outcomes even when controlling for their methylation subtype and also for WHO grade. However, prior grouped analyses demonstrated or suggested that heterozygous loss could also portend poor prognosis. So what did we do? We used DNA methylation data to assess a set of 776 meningiomas, including a cohort of 659 for which we had clinical data. And we assessed the CDKN2A and B status using copy number analysis. You can see some of our sample traces here, which we generated using our methylation data and then manually assessed, grading them as homozygous or heterozygous loss by the loss of, or the degree of loss of the copy number. So what did we find? In our entire cohort, we found that 4.9% of tumors had any type of CDKN2A and or B loss, and that these losses were very concordant in their zygosity, such that if CDKN2B had a heterozygous loss, CDKN2A would also have a heterozygous loss, and same thing for homozygous losses. There were 3 tumors in which there was heterozygous loss of CDKN2B with homozygous loss of CDKN2A. The remaining clinical analyses focused on these tumors that are highlighted in red that had concordant loss. The mechanism of the loss, we discovered a little bit that the majority of these tumors that have CDKN2A and or B loss are caused by loss of the entire arm of 9p chromosome. But there was a substantial minority that did have just focal gene deletions. Now, the main takeaway here is that heterozygous loss, just like homozygous loss, also predicts worse recurrence-free survival. You can see in our entire clinical cohort that if you had intact CDKN2A and or B status, the median recurrence-free survival was not even reached. Whereas for heterozygous loss, the median recurrence-free survival was 20 months, and for homozygous, 10.5 months. The difference between homozygous and heterozygous in this cohort did not reach statistical significance, but there was certainly a trend that homozygous may be worse than heterozygous, suggesting possibly a gene dose effect. Looking at our other clinical factors that we assessed, we saw that there was an increased preponderance of WHO grade 2 and 3 in tumors with any type of CDKN2A and or B loss. There was a stepwise increase in the MIB or the Ki67, such that intact tumors had the least and homozygous tumors had the highest, with an intervening middle ground in the heterozygous tumors. The difference here between heterozygous and homozygous tumors was statistically significant. We saw a male preponderance in the patients who had a loss of CDKN2A and B, and that these tumors were more likely to be recurrent tumors. Now, we did not see a major statistically significant difference in the extent of resection. We did control for WHO grade as well as only looking at primary tumors, and these trends remained the same. Those tumors with CDKN2A and or B loss within any of these cohorts did worse than those without the same gene loss. So that addresses the first point. Second point that we'd like to make is that these losses occur only almost entirely in the MENG-C aggressive tumors. So previously, we've published a classification of the MENG grouping, A, B, and C. A tumors have intact NF2 status and are normally benign. B have loss of NF2, but don't have the same copy number instability that we'll see in the aggressive group C tumors that are characterized by loss of 22q as well as loss of the chromosome 1p. You can see here, this is all demonstrated in this oncoprint that shows that almost all of the CDKN2A and or B losses were observed in the MENG-C group, which is designated here in red. Only one tumor that had CDKN2A and or B loss was outside of this group. Of these tumors that had CDKN2A and or B loss, all had concomitant loss of chromosome 1p, and all but 3 had concomitant loss of chromosome 22q, suggesting that these tumors are the most arranged in an already aggressive subset of tumors. Still, even within this aggressive molecular subtype, MENG group C, the CDKN2A and or B status predicts recurrence-free survival, such that those with any type of CDKN loss do much worse than those with intact CDKN status, even within our aggressive meningioma group C. So the major conclusions for this study are that the heterozygous loss of CDKN2A and or B is similar in outcome to a homozygous loss and should be considered in the next iteration of the WHO grading schema. We are a little bit limited in that we don't assess the protein product of the CDKN2A and or B, and there is, at least historically in the literature, not a one-to-one relationship with the copy number and the protein expression. Similarly, we don't identify or search for pathogenic mutations that could lead to gene loss that would not be detected by these copy number counts, and these have been previously reported. So our 4.9% number may be slightly underreporting. We do identify that these losses occur in already the molecularly aggressive subtype of tumors, suggesting that these may be the most deranged of that subtype. And regardless of the classification system, CDKN2A and or B loss ultimately is a very strong predictor of poor recurrence-free survival in these meningiomas. And with that, I'll conclude. Feel free to reach out to me. Thank you.